Would you like to introduce your students?
For sure. So my name's Dr. Sarah Wofford. I'm in the biology department. I have a few different posters that my students will be presenting today. So we've recently redone our labs. Specifically, this is from the general ecology lab, to design them in such a way that students will work under a theme to ask certain questions and then develop their own experiments, carry those out through the semester, and then analyze that data. So for this last semester, our main theme that we were working underneath was urban heat island effects, or the impacts of microclimate shifts due to urbanization. So for the first group that's going to present their work, they specifically looked at the instances of coliform bacteria, and I'll let you guys take it away.
So our research was on urban heat island effects on coliform bacteria growth in Lotic systems. First thing people ask is, what does Lotic mean? And the thing that we decided to focus on is urban heat island is a phenomenon where the temperatures in urban areas are higher than the temperatures in rural areas. And this is due to solar radiation being absorbed by concrete, by asphalt, roofs, other things. And so it creates this pocket climate to where in that area temperatures are significantly higher than the surrounding areas. And so this is a proven fact in temperature. And one thing that wasn't really studied was the effects on aquatic systems. And we really focus on Lotic systems, which is flowing systems, rivers, things like that. And so we decided that we would test to see what happened to the bacteria in these different areas. The bacteria that we chose was coliform bacteria, and that's E. coli, things like that, things mostly found in the stomach that cause problems. And the reason we chose coliform bacteria is because it is a good indicator of other harmful pathogens. Some of these pathogens are small and they're not easily detectable, but coliform bacteria is usually present in larger numbers if the more harmful ones are still there. And so we decided that we would test those, and we hypothesized that the warmer, the more urbanized the system, the more coliform bacteria we should see. And the reason that we decided to do this is because we really wanted to focus on what is our impact, or what is the impact of these urban heat islands on human safety, on human health. We wanted to focus on it to see if, did this cause problems for specifically urban areas, because there were so many people there. So I'll turn it over to Kelsea for our methods.
In the field, we went to two different locations. We chose a rural location and an urbanized location. For our rural, we chose Terrapin Creek, which is toward Piedmont. It's very rural. And for our urbanized area, we chose Logan Martin. And you can see by the pictures how different they are. Like, here's a bridge and it's over a highway, and then at Terrapin it's more mossy and not many people. And I actually went to the Logan Martin location and took the samples, and there was people kayaking, fishing. It's very populated. And so we used the WorldPack collection bags, which are shown right here, and we took 10 samples from each location, and we measured out per location, took the coordinates and the temperatures of the locations of the samples. We labeled them, took them back to the lab, and we plated them using the Skin Easy Gel. And what we did is we would open the solution and take the samples and put that in there, shake it up, and then plate it. And after 48 hours, we let them incubate. We went to the plates and counted the colonies of coliform on each plate. And you can see in our t-test that we ran the difference in colony counts. Like, here is the Terrapin versus the Logan Martin. And Logan Martin was much higher, and so were the temperatures. And we compared them to a control, like we did DI water for our control of, completely sterile, and then E. coli for our other control, and we just compared how they would look versus, the colonies and stuff. But in our t-test, we just related it between water temperature and E. coli counts and colony counts, and the temperatures were higher and the colony counts were higher in the urbanized area.
So just to sum everything up, and the reason why we did this was, like Quinton said in the beginning, it affects humans, and this is pretty populated areas that a lot of people go to. And E. coli, like you said, is an indicator for pathogens, and specifically like salmonella and things like that. And I've done some extra research on this. E. coli, there's a few strands that are starting to build resistance to antibiotics, and what are the chances of them maybe spreading that like resistance DNA and stuff like that to salmonella? And we get to the point people catch that, and maybe we're not able to cure it completely, or not cure it, well, make them not get sick. And that's just a big point of this. It's just see like how much it's going to affect us.
We accepted our hypothesis in the end, concluding that urban areas do have greater bacteria growth, which could be dangerous for people. And I think if we could do further research, we would probably look at a more urbanized area just to get a bigger feel of really how it would work in a larger urban area. So there's plenty more that we could do with this research, but this is just getting our foot in the door. So.
And like the next step we could do is maybe fecal contamination, like how much of that's being our water source, instead of just focusing on heat, like what's being dumped into the systems that could also help the growth of E. coli.